Bismillahir Rahmanir Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Peace be upon you all, and welcome to another live session of our evolution course. And today we are going to go through the very exciting process of the transcription of DNA. In the previous episodes, we have already gone through. Actually, this is the third part of the central dogma of biology. And during the first parts, we have made this analogy, if you remember, between the cell and a computer system. And we said that a cell, the genetic system, we can make this 10-step analogy of how it functions. And you need to probably review those two previous episodes, but you can still watch this one if you have not watched them. So this was part one. You can look it up. And then there was part two last time, which looked like this. And we have gone already through 5 pieces of the analogy, starting from the DNA. The first one was the DNA as a storage system. So we have gone through that. And then we explained how its replication is like copying data. And then we explained how the epigenetic system is like an encryption/access control system. And then we have shown how the system actually repairs itself, repairs damage, identifies and corrects errors that happen during the replication process, or actually damage that happens during the environmental process. So this is a system that does not just haphazardly work. It is a system that works to do faithful copying of itself to preserve life. And it is actually very funny how evolutionary biologists claim that evolution happens through accumulation of errors. And we have had a live session after the last episode, and we pondered upon that. And then the last thing we explained last time was the nuclear pores, those little holes that are in the nucleus that act like firewalls that protect actually the genetic system from being, let's say, hacked. And we've seen how nothing gets inside the cell or goes outside of the cell, except after being checked by those genetic pores to make sure that the right stuff is coming in, and what is allowed to go out goes out. So this is like a fingerprint, a password, a firewall that protects the system from being hacked. Now, today we are going through the DNA transcription. We have seen how DNA has this structure that helps it condense and compact data on its strands in chromatin. And why is all of this happening? It is happening so that DNA can eventually deliver the information that is stored into it to the system to produce what this information represents. So this information eventually translates into products like RNA and proteins. And the first step in this process is what is called transcription. So what does transcription mean? Transcription means reading the DNA material and converting it to RNA. And since we have already made the analogy that DNA is like a storage system for the information, RNA is essentially like the same program that is stored, or data that is stored on the hard disk, but now it is loaded into memory. So DNA is like data stored on a hard drive or a tape drive, and RNA is like programs and data stored in memory. So now we need to read the data from the hard drive and load it into memory. And then this information, or this program, when it's loaded into memory, it is compiled and then it is rendered into the final product. So today we are going to go through this step of transcription. So again, what does transcription mean? Because you will have quizzes, now if you're not subscribed already to Evolution course, you need to go to the website. And when you go to the website, I'll put it here on the ticker. So you subscribe to the course, and then you can go to Article 8. And in Article 8, you will find a compilation of all of this. So and then there will be the quiz. So once we finish all the parts, I will be putting the quiz so that you can answer the quiz and you make sure that you have understood all what we have gone through. So transcription means reading DNA material, converting it into RNA. So the first thing we need to understand is what is actually stored on DNA material. What is on the DNA material is what we call codons. And codons are three individual pieces of information. Each piece of information is like a bit. You know, in the binary code, you have a bit that can be a 0 or a 1. The DNA code does not use 0 or 1. It uses one of 4 alternatives. So it's as if 0, 1, 2, or 3. But instead of this, it is actually 4 letters, A, T, C, or G. Okay, so you have 3 locations. Each one can be one of 4 possibilities. And all 3 together are called a codon. And one of them only is a nucleotide. And since DNA is 2 pairs, so 2 nucleotides looking at each other, they are always in complementary pairs, is called a base pair. So all those 3, when they are read by the system that we are going to see, they are going to be first translated into RNA, and then RNA moves into proteins. Each one of those 3, each piece of it can be one of 4. If you do the calculation, you will find that this can produce different 64 arrangements. So you can have, for example, AAA, AAT, ATT, TTT, and so on and so forth. Those different arrangements can go up to 64. So we can expect that there are 64 products out of each 64 different products out of each different codon. But the reality is that we don't have 64, we have less than that because there is redundancy. So whenever we look into the system, and this redundancy causes that if there is an error, since there are 64 possibilities, there are only 20, a little bit more than 20 possible products. So it means that there is almost 1 to 3 each on the average. So if there is one change to one of the nucleotides, the final product might actually be the same, because maybe the first and second and third arrangement, if just one nucleotide changes, it will give the same. To understand how this works, I will show you what is called the DNA codon table. So this is the table. It can come into different configurations. One of the nice and fun configurations is the DNA codon wheel. So let's look at the center here. We can find at the center the first letter. So if the first letter is a G, the second letter can be, for example, if you look at the example, a T, a C, an A, or a G. And then the third letter, if you go G and then T, then it can also be a T, a C, an A, or a G. And then in the outer rim, you will find what is the product. And you will find, for example, if you start from 12 o'clock, you will find that TTT or TTC can give the product called F. And F is like, stands for a certain protein. And then you can find TTA or TTG can give the product L. So in this case, for example, if TTT changes to TTC due to a mutation, we call this a synonymous mutation because it still produces F. So the system is built in a way so that it is resilient even if after all the error correction and damage repair, etc., 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 that we have looked at before, the system is still resilient to errors, which is essentially amazing, because if evolution depends on errors, then how come the system resists errors by all those layer after layer after layer after layer of resisting errors? And obviously, it is because errors cause damage and harm the creature and can cause disease or deformation or disfigurement or disability or death. one example, we can notice here that there are 4 letters, U, C, A, G, while there we had T, C, A, G, because the letter T is changed into a letter U when it goes to RNA, and RNA is actually what is translated into proteins. So when you look at here, if the first position is U, the second position is U, and the third position is U or C, then it is phenylalanine, for example, the protein phenylalanine. If the third letter is A or G, then it is leucine, for example. So you can find here that leucine is produced by 1, 2, 3, 4, 5 different possibilities, where the first letter can be a U or a C, and the second position needs to be always a U, and the third position can also change according to that. So this is the resilience of the system. And we can see the same genetic wheel based on RNA. So now we understand what needs to happen. The first thing is to convert these letters that are of DNA letters into this, which are RNA letters, and then the RNA letters will form codons, and then codons will be translated into proteins after splicing. So this is what we are going to see. This is plenty of information. So we will still have a 4th and maybe a 5th part to complete this. But let's remember that the objective of this course is not just to make you know that evolution does not work, but it is to equip you with the information so that you can explain to your children that this matter of randomness is completely nonsense. Because when you are going to see what we will see today, plus what we have seen before, plus what you will see later, you will totally understand that this is a system that is amazingly engineered to the extent it is impossible that it is there by any way of randomness or chance. The first thing that we need to understand is now we have the DNA. The DNA will be transcribed. So we need to think, what will make it get transcribed? So DNA is sitting there in peace in the nucleus, and we need a protein to be produced. How will it understand that it needs to be transcribed? So let's see this video, and in the video we will see that there is a signaling system. There are 2 types of transcription. Transcription that's happening all the time, because, for example, it needs to make a product that is related to metabolism, and metabolism is happening all the time. So the metabolic machinery is being produced all the time. Metabolism is what causes us to convert food into energy, for example, and many other processes in the cell. And there are proteins and products that are produced through the DNA transcription translation process that need to happen or to occur on demand. Also, those transcripts are different. So the kinds of proteins that need to be produced in muscle cells is different from brain cells, different from liver cells, etc. So the transcription process needs to be triggered, number 1, according to the type of the cell, and number 2, according to the current needs, the current context. So this first video will be something that gives us an eye-opener about how this process is regulated, and let's have a look.
mRNA and eventually proteins are produced when and where they are needed in an organism. Some genes, such as those for housekeeping proteins in processes like glycolysis, are expressed all of the time. These genes undergo what is known as constitutive transcription. Other proteins are only produced at specific times in certain cells. In these cells, genes undergo a process called regulated transcription. Transcription is regulated by proteins called transcription factors. These factors are proteins that are produced in the cytoplasm and eventually migrate into the nucleus, where they interact with DNA and activate transcription. These transcription factors only interact with specific genes, those genes whose transcription they control. Most eukaryotic species have over 1,000 transcription factors.
1,000 transcription
Regulated
factors over
transcription
that.
begins when a signal is received by the cell. This signal, often a protein, begins a signal transduction cascade that lets the cell know certain proteins are now needed. The most common activation
now
event
knows.
for a protein in a signal pathway is the addition of a phosphate group. That activated protein in turn interacts with another protein. This interaction leads to the phosphorylation of the next protein in the pathway. There is often a series of proteins that are activated by phosphorylation. At the end of this cascade, the final protein will be activated and then enter the nucleus.
Remember the nuclear
It must
pores?
make this journey because transcription only occurs inside the nucleus. After entering through a nuclear pore, the protein interacts with the specific transcription factor responsible for activating mRNA production. As with the earlier proteins, the transcription factor is modified through phosphorylation. The activated transcription factor will next bind to an enhancer region. The enhancer is a region
Remember
of
this
DNA
enhancer region
upstream
for
of the
later.
transcription start site that binds a transcription factor. The transcription factor then binds to the DNA and moves to interact with the rest of the transcription protein complex, located at the transcription start site.
All of this
When the
has come from
formation of this
outside,
complex is
assembled.
complete, transcription
to
of the
start
gene
the
will
process.
begin. It is this sequence of events that ensures genes required for a specific tissue or only at a specific time are expressed appropriately.
All right, so I had a comment that some echo happens, so I'm going to mute while showing the video, the next one. But now let's think of what we have seen. So a signal comes from outside of the cell telling the cell, dear cell, you need to produce this protein. And this creates a signal cascade. The signal cascade goes from one step to the other until essentially a protein complex is formed. It gets, for sure, the password that it will need to use so that the cellular pore will let it in. So there is plenty of intelligence happening outside the cell. We didn't reach to the DNA itself. And this makes us think of what is really controlling the whole thing. What we really know on the ground about how this thing overall works is really minuscule. And then this cascade happens. This cascade needs to happen in a complete way so that at the end you have the right pieces that have the right password on them so that they can be let in through the nuclear pores. Nothing happening at random there at all. And then they go to the... Now we need to think, these are just molecules. These are just dumb molecules, consciousness molecules. How do they know, even after the protein cascade all happens, that they need to go towards the nucleus and exactly at the nuclear pore and pass inside? Nothing really by accident there. So we need to really think very well and get our attention drawn to the fact that this whole thing is 100% designed to produce this effect. And when you see a series of events, any one of them that gets blocked destroys the whole system, you understand the irreducible complexity of this system. Because it cannot work except if it is all of it present as a system. So can this really evolve? The nucleus with the nuclear pores, with the signal that comes from the outside, so many details how this signal will knock on the door of the cell and be let in, because there are mechanisms also at the surface of the cell that identifies this signal and delivers a signal internal to the cell, and then the protein phosphorylation cascade that eventually produces the product that needs to go inside. It is passed through the pore, and after the pore inside the nucleus, it gets assembled in a certain way so that there is a piece of it sitting at the enhancer and a piece of it sitting at the promoter, and then they go and join together to start. We're going to see all of this. I want you, while looking through it, to contemplate on this, whether it is by any means possible that this is happening or could have been a product of a mutation, a random mutation process acting upon by natural selection. Can this system have occurred like this? So the next thing that we are going to see is
the central dogma of molecular.
The next thing that we are going to see is to have a little focus on the transcription process. We have seen how those products come in, and a piece of them stands at the enhancer region, and another piece stands at the promoter region. Now, transcription needs to start, so we are going now to have a deeper look at what is called the transcription initiation process. Actually, we are going first to see, the whole transcription process, and then we are going to focus on the initiation process. So this one is a simplified version, and then we go into further details, into the initiation, and then into the promoter region itself, and then into what happens into the polymerization step, and then what happens inside polymerase itself. This will be a great ride, and then we will go into the Q&A. So if you have questions, write them down, or write them on the chat, and then I will answer the questions in the Q&A session. And I have pinned the Q&A session link to the live chat, so you can go there and move to the and set your reminder bell or something. Okay, so let's have a look on the overall process, and then consider and then focus on each part of it.
In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription. Or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
So I said we are going to look at the whole transcription process, and I was wrong. I prepared this video in the middle so that to ponder on this amazing scanning process, because in the first video we have seen the transcription factors coming in. One just dropped in the enhancer region and another one in the promoter region, but this is essentially not what happens. And the more scientists learn about how crazily complex this process is, the more we feel how small our knowledge is to the reality of what's happening. Remember when we have just said, how do those proteins know they need to go into the nucleus and go to the pores? Because essentially there are mechanisms also to guide, whether we understand the mechanisms or we don't. There are crazily sophisticated mechanisms like this that you have seen the transcription factor doing the scan, and it knows what it's looking for. It seems so. And it jumps from one strand to another strand until it finds the right place and it stops there. And you see how it hops, and what kind of complicated composition of this nanorobot that needs to be there from those hydrocarbons to the extent it can perform this function. What amazing design needs to be put if you are to design this nanorobot that just does one small step of this process in an entirety of small steps. Each of them is amazingly complex. What kind of engineering skills you need? So now let's have a look at what we have seen now how the transcription factors bind to DNA. Now we'll have a look at the overall process of transcription from DNA into RNA in a simple thing, and then we do further focusing.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA Polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
Wow, so this was the journey from transcribing the DNA into producing the RNA, splicing or cutting parts of the RNA, and then having this final thing called the messenger RNA that flows outside of the nucleus. Obviously, it's not as simple as it looks. There are a huge amount of complications. And obviously, after the mRNA is produced, it is translated into proteins through the ribosomal complex, but we did not see that in this video. Because there is so much details in the transcription process that we'll almost spend the whole of today on it. So now let's have a look at how this transcription starts. We have seen the components being arranged in the cytoplasm, going into the nuclear pore, meeting other components inside, and then a part goes to the enhancer, a part goes to the promoter, and then we have seen how the overall thing. Let's now see how the process actually, the details and the intricacies of how initiation of transcription works.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA.
This is the promoter part.
Following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA Polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated.
It's really a complex.
The
now look at
initiation
how
complex requires contact with activator proteins, which bind to
remember
specific
this guy
sequences of DNA known as enhancer regions.
This
These
part was
regions
sitting
may
in
be
the enhancer
thousands
region.
of base pairs distant from the start of the gene. Contact between the activator
Look at the roller
proteins and
coaster
the
effect.
initiation complex releases the copying mechanism.
Now this is the
RNA polymerase unzips a small portion of the DNA helix,
transcription
exposing the
happening.
bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand.
Look how it collects the small letters from the
Subunits can be seen
input.
here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Now the RNA has been produced. So I'm using the speakers to avoid the echo. But if you are not amazed by this, what will make you amazed? If this doesn't knock you off immediately as an amazing machine inside, and this is happening every single moment, not every second, every microsecond, every nanosecond, this is happening. Continues to happen all the time, otherwise we will die. Transcription after transcription, RNA production after RNA production. See how the RNA transcription process, the DNA transcription process into RNA started by this complex that arranged itself in the promoter region. And then the other piece that was sitting in the enhancer region that both came in, and what we have seen when they came through the pore and arranged themselves. And then the enhancer region comes and touches the promoter region, and then something else happens to provide energy, and then woo, it runs like a roller coaster. What level of complexity? And to further appreciate the complexity of what happened at the promoter, let's look more into the further details of what really happens at the promoter. And because, you know, the more you dig into this, you find details that are more amazing, that tells you that those are very intricately designed machines. Let's have a look at this. This is what happens just at the promoter to start the process.
Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box.
Tata box is just
Enhancer region may also
another
be
name.
involved.
Several
Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach.
How do those guys know they need to come now?
These complexes prepare the DNA for the successful binding of RNA polymerase.
So this RNA polymerase is what is going to run as the roller coaster later, okay, and do the transcription
Once
process.
RNA polymerase is bound, other transcription factors complete the mature transcription complex.
Now we need some energy, you know. See the transcription complex, the polymerase,
Now,
all
energy must
the
be added
pieces
to the
coming together.
system for
Here is the
transcription
energy.
to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates, and the newly formed strand of RNA is released.
Wow. Now you might also ask, how does it know that it ends? Well, it knows that it ends because there is a codon sequence that is called a stop sequence. And when the RNA polymerase sees the stop sequence, it stops. How amazing is that? And this thing, which is the DNA that has been transcribed, starting at the promoter, ending at the stop, is what you need. If it's more or less, the production, that whatever is produced will be rubbish. Okay, we'll work on the echo by completely muting next video. All right. Now, you have seen the machine that is being assembled at the promoter region and how the enhancer comes to it, and then the rest of the pieces come to it, and then the energy comes to it. What is guiding all of those components to come here? What is guiding them is even much, much more sophisticated underlying processes. So it is a huge pyramid of complexity after complexity after complexity after complexity. And the more we know that we do not know. This is the problem of this system. And when you go into the literature, they will tell you evolution did it. This is not an answer. Evolution did it is not an answer, because evolution did it is irrational in the first place. These are designed machines. And that is why evolutionary biologists, etc., they will not show you this in their evolution nonsense videos. because once this is publicized in the proper way, okay, other than the random mutations did it nonsense, nobody will believe it. And this is not the end of it. We have just seen a little bit of how the complexity of just starting the process. Okay, just starting the process. Now let's have a look on this thing that is running as the roller coaster, the RNA polymerase. Let's have a look. What is it and how? You have seen the components coming, rushing into its intake, and then inside it being arranged, and then outside of it goes out, ejected is the RNA strand. What is this machine? This is just one of the tens of machines that you have seen involved in the process, but this is the one that does the copying. Actually does the transcription, copies one of the strands of the DNA into an RNA strand. Just to give you an idea about those blocks that look like Lego blocks, are they just really like those blocks? No, they are very complicated things. Let's have a look in this thing, into what it really is.
Eukaryotic RNA polymerase II transcribes all messenger RNA molecules in the cell. It is a huge complex of 10 different protein subunits. The active site of the enzyme lies at the interface between the two largest subunits. In this structure, a short stretch of a DNA-RNA heteroduplex was co-crystallized. New nucleotides would be continually added to the 3' hydroxyl group of the RNA shown in red. In the active site of RNA polymerase II, a single-stranded DNA template is transcribed into a complementary RNA transcript. The initial product is a DNA-RNA hybrid from which the newly synthesized RNA strand is stripped off. It leaves the polymerase via an exit groove on the protein surface. A pore on the backside of RNA polymerase II extends from the protein surface all the way to the active site. The nucleotide triphosphates used to build the growing RNA transcript enter the polymerase through this pore.
Wow, so this was this thing, this complex called RNA polymerase. And what it is really more than 10 protein molecules attached together. And each one of them is this mesh that you have seen. And this mesh is essentially, what we will see later, is a protein that used to be a strand like the RNA, but it gets folded and reattached to each other in what we'll see as the protein folding process. So just one linear strand becomes 3D structure, and then those 3D structures get attached together to produce this complex. And see how there is nothing left to chance. This meshed thing has a place that is an intake that goes all the way so that nucleotides will come in, and then they will go to the binding sites where they are going to be arranged, binding each correct nucleotide that is going to be piece of an RNA strand with the correct nucleotide of the DNA strand, replicating and copying. This is just one of the components that are used, and you have seen how many components are there in this process. And you can also ponder, this complex is made of proteins. But to produce proteins, you need the DNA to be transcribed to RNA, and then RNA to be excised and spliced, and then the mature mRNA is then translated into protein. But to have this process already working, you need to have proteins. So, which is one more of the so many paradoxes that can only be solved if this system arrives together as a complete system. And in the earlier episodes of this course, the earlier sessions, I have deconstructed the nonsense called RNA world hypothesis that says that maybe everything started as RNA. And if you didn't watch those episodes, please go back and watch them, because when you go into evolutionary literature, they say, okay, maybe this happened after the evolution from the RNA world, etc. And yeah, chicken Yeah, man's man says chicken or egg. It has to be chicken and egg. Yes, it has. It cannot be. The protein was there before DNA, because we know that you need DNA then RNA to produce protein. And the whole theory, if you consider that this thing evolved through random mutation, it's completely impossible. Now let's even have a further look on a further complexity. I will try to finish this before the hour, so that we start the Q&A session at 12:30. Okay, so we only have like 13 minutes, so we'll probably just finish the transcription today. And just remember, if you have questions too. So now we will look more inside what happens into this protein complex, inside the polymerase. Let's look at what's happening inside the polymerase, that we have seen its composition. We will start from the point that it starts working, and then the roller coaster, and what happens inside this roller coaster really is even more amazing.
To produce a functional RNA molecule, the cell must make an RNA copy of a DNA sequence. In double-stranded DNA, the strand to be copied is known as the coding strand. The other strand, which contains the complementary base sequence, is the template strand that will be used to form the RNA transcript. So what are the key features of the coding strand? The start of the coding sequence is known as the transcription start site. To the 5' side of this site is the promoter region. The promoter region contains particular DNA sites known as core promoter elements. These are important in the assembly of the complex of proteins that are needed to initiate transcription. This complex is called the Transcription Initiation Complex and contains the RNA Polymerase II enzyme along with additional proteins or protein complexes known as transcription factors. Transcription factors help RNA polymerase to locate the promoter and initiate transcription. We'll now consider how the initiation complex assembles. The first step in the assembly of the transcription initiation complex is the binding of the transcription factor TF2D to the Tatar box, which is one of the core.
This is like a review of what we have gone through, but the important piece is when it starts explaining what's happening inside the polymerase.
This key subunit is known as Tatar binding protein, or TBP. The TBP subunit induces profound bending of the DNA. Binding of TF2D is followed by the binding of TF2A and TF2B. TF2B binds to the core promoter element called the BRE, as well as contacting TF2D. In the next step, the RNA Polymerase II core enzyme is recruited, together with TF2F. Finally,
here is the polymerase.
TF2E and TF2H are recruited to form the complete transcription initiation complex. This is the
I like it
closed
when they use the
complex,
term recruited,
meaning that the DNA is still double-stranded and the template strand has not yet been exposed. Transcription begins with a separation of the two DNA strands to form the open complex.
Look at that.
This separation depends on TF2H, which uses energy from ATP hydrolysis to unwind the DNA and promote strand opening. The short stretch of DNA with unpaired strands is called a transcription bubble. The opening of the transcription bubble exposes the template strand, which will be used to form a complementary RNA strand. 3 ribonucleoside triphosphates enter through the funnel region of the RNA polymerase and form base pairing interactions with the template strand. RNA polymerase catalyzes the addition of nucleotides to the 3' hydroxyl of the last base in the growing chain. This means
This is what's
that
happening
RNA
inside
grows in the 5' to 3' direction. Transcription initiation does not go smoothly. RNA polymerase typically will start to synthesize the RNA, but the RNA product will diffuse away after it has reached a length of just a few base pairs. The whole process of synthesis then starts again. This process is known as abortive initiation. After multiple rounds of abortive initiation, RNA polymerase breaks free of the promoter and continues to synthesize the RNA in a more persistent way. This phase is called promoter clearance. RNA polymerase now forms a stable complex with the DNA and continues to synthesize the RNA. At the same time, TF2H phosphorylates the C-terminal domain of the large subunits of RNA polymerase II. This domain is called CTD. This phosphorylation of CTD is key to the processing of the RNA to produce the mature transcript, and this is what happens next. During the RNA processing step, a complex of RNA processing enzymes called PTEFB binds to the phosphorylated CTD. As the 5' end of the growing RNA comes out of the exit channel of RNA polymerase, PTEFB covalently attaches a guanosine cap to the 5' end of the RNA. PTEFB then attaches additional phosphate groups to the CTD, which causes transcription elongation, which is paused to resume. The more highly phosphorylated CTD also can recruit additional RNA processing enzymes that will be needed to complete processing of the transcript at the end of the transcription cycle. RNA polymerase has now transitioned into the elongation phase. During this phase, the growing RNA strand comes out of the RNA polymerase exit channel as it continues to elongate. Transcription bubble moves along with the RNA polymerase. DNA base pairs are separated ahead of the transcribing polymerase to expose more of the template strand, while bases in the wake of the polymerase re-anneal. In this way, RNA polymerase can continue until it has transcribed the complete RNA. RNA polymerase doesn't always make it to the end of the coding sequence in one smooth journey. Instead, RNA polymerase can sometimes stall before reaching the end of the transcribed region. The polymerase can be restarted by the binding of elongation factors, but sometimes the RNA polymerase starts to backtrack. This means that the polymerase slides backwards along with the transcription bubble. This causes the 3' end of the growing transcript to become unpaired and to come out through the funnel region of the RNA polymerase. The transcription factor TF2S can rescue the stalled polymerase by binding in the funnel region and promoting cleavage of the 3' end of the RNA that is unpaired. This exposes a new 3' hydroxyl in the RNA that is correctly positioned in the active site. Transcription can now resume as before. Transcription continues until RNA polymerase II reaches a sequence in the DNA called a polyadenylation signal. After RNA polymerase is transcribed through this signal, sequences in the RNA transcript associate with the RNA processing proteins that are bound to the polymerase CTD. These proteins cleave the RNA transcript and add a sequence of adenine nucleotides to the 3' end of the transcript. This run of adenines is called a poly-A tail. Once the poly-A tail is added, RNA polymerase dissociates from the DNA and the mature RNA is released. At this point, transcription is terminated and an RNA transcript has been successfully produced.
So I got a comment that is essentially really expressing my own feelings. So Ilyas Al-Modi says, this is tear-inducing, unbelievable, all of this in every cell countless times. Yes, all of this in every cell is like thousands of it happening at any moment. And there are trillions of cells. And this is happening all the time. And just look at this process and look at how unbelievably detailed, and there is a corrective mechanism for everything. This is something that's happening in a fluid in a very small place that is stuffed with all those pieces, and there is a machine that will fix every possible scenario. It moves. If it gets stuck, it recruits. And they say it recruits. Come on, guys. What is essentially recruiting? This is a designed process. And it just happens that if it stops, something comes. Energy is being poured. If it stops and backtracks, something, another piece comes and cuts, because the backtracking will cause the output to go from to go back to the other side. So it cuts it and exposes a new piece, and so it attaches again to the DNA, so that when it goes forward, it is correct. And by the way, there are even quality assurance processes that if the RNA that is produced for any reason is defective, something will go and check it and degrade it. There are so many details, and each of those, how does those components happen to go there, and they get recruited and they reach? Remember the second video that we saw about how the transcription factor will continue scanning until it finds the correct place, and it recruits again other pieces so that they help it bind, etc. Every single detail has a pyramid of details underneath it. Pyramids upon pyramids upon pyramids upon pyramids. Unbelievable complexity. And the slightest mistake and the slightest missing piece will cause the whole thing. Only the blind-hearted and blind-minded and blind-eyed cannot see that this is a superiorly and magnificently engineered process. Mansman says, imagine if we had to consciously manage all this. Yeah. Imagine if we needed to design. We cannot design 0.0001% of this complexity. This is superior design. This is a message for those who do not understand what is happening in the cell, and they say that this might have happened through randomness. Okay? And for those who understand and are hiding the truth from people, show them the science and let them make their mind. Don't just throw the nonsense at them. And yeah. This was I'm happy that this has induced the right feeling, because and let me tell you something, this RNA strand that came away now is the data and the program that has been read, and it goes into the memory. There isn't enough time to show the splicing, but I'll just show you an introduction to the splicing. An introduction to the splicing. There's another comment that says, and this is just an adaptation, who knows what it actually looks like. Remember, Darwin thought it was a blob of jelly. Yes, absolutely. Not a blob of jelly. It's a blob of an amazingly complex, electromechanically engineered, chemically executed, biochemically executed process of unbelievable sophistication, unbelievable engineering sophistication. Let me just show you what happens to this, and this is an introduction to the splicing, and the splicing itself will probably take the whole of the next session. Just show you what splicing is. So this RNA strand that is produced now is the program and the data that have been loaded that need to produce something else, or to actually become an RNA on its own, because RNA on its own can be used as a machine, like when we would see the ribosomes. So this RNA strand needs actually to be compiled. Pieces of it need to be cut off, and other pieces need to stay, and the pieces that are cut off are called introns, and those that are going to stay are called are called exons. And the introns can be removed, and then the exons can be stitched in different configurations. So the same RNA strand can actually produce different products according to this unbelievably amazing process that is called the splicing that we are going to see now. I'll just show you this, and we'll probably conclude and go to the Q&A session right after. So here we go.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Now, what do you think of that? If you are not holding your tears now, you should really examine your heart. I have no other comment. And this RNA that has moved outside of through the nuclear pore, there is a spliceosome waiting for it. It assembles, and the machine understands that there are introns and there are exons, and it excises. It takes out the introns and forms them into a loop, and then stitches together the introns. And guess what? There are different configurations of stitching the introns, and it will perform the correct configuration according to the needed protein. And we don't understand why or how or on what basis it's doing that, but we are working and we are living. And guess what? The evolution, the genius evolutionary biologists thought that introns were junk. Just like the usual thing they do, they think everything is junk because they thought they want to sell you that this is random, while it is not. And then as time goes by, we understand that introns have essential functions, and that those loops of introns that are excised, cut off, spliced off, will then be shaped into RNA complexes that have functions. And without those functions, we will not be living. Unbelievable complexity, unbelievable, and we understand that much of it. So small piece of a huge mountain that we do not know where it ends and where it starts. And then they tell you it's random. Yes, Big Man says this cannot be random. Absolutely, my friend, it cannot be random. Yeah, Elias says, oh my God. Subhanallah. Yeah, and Elias says, I have to share this with my family. Please, when I saw this, I did share it with my family. And I told them, this is bringing tears to someone's eyes. Look at this. And imagine that this is happening every single instant. Trillions of times, every single instance. In your body, there are 30 to 40 trillion cells inside each of them. Inside each of them, this is happening hundreds of thousands of times as we are speaking. Otherwise, you will die. Can you believe this? Thousands of trillions of times this is happening every single moment. And Allah is maintaining us and keeping us alive. All this unbelievable complexity is happening inside us. We need to say Alhamdulillah. We need to say Subhanallah. We need to tell everyone who says that this is random, get out of here. We are not crazy to think that this is random. Anyway, so I meet you again next Saturday, and please join me for an open discussion on the Q&A. I will also invite those who would like to come on board with me on the platform to tell their opinions and share their thoughts or have questions, and we'll have a nice discussion like last time. So Subhanallah, Alhamdulillah, La ilaha illallah, Subhanallah wa bihamdik, la ilaha illa anta, astaghfiruka wa atubu ilayk. وعصر الإنسان الذي خسر الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. I will initiate the Q&A now while I'm putting the extra here. Please join me on the other side. I hope you have made use of this. Please share this with your family and ask them to subscribe to this course because it fixes one's heart to see this. Please do this and share this to the end of the earth, and let's glorify Allah subhanahu wa ta'ala, and I'll be waiting for you on the other side shortly. I will be putting on the other live stream now because and I will be putting the extra. Join me now on the other side, and peace be upon you all. Assalamu alaikum wa rahmatullah. You find the link to the other live stream on pinned to the live chat.